Hi, this is Dr. Ben Morrill. Welcome to episode 99 of Reptile Genetics Weekly. Happy to have you here. Going to be a fun one today. Get to talk about a whole bunch of stuff we haven't really touched on before, so that'll be fun. Oh, yes, and also we did hit 3,000 subscribers on YouTube, so thank you to all of you. Thank you for the likes, the comments, the subscribing, spreading the word either directly or at least by, hitting a notification bell or however you do it. All of those things we appreciate. That helps make sure that this gets out to more people. So if you're watching this and you're new, please do those things. If you've been here for a while and there's anything that you appreciate we're doing, at least give us a like or follow or something. We really appreciate that. But 3,000, we're very happy with that. And like we've talked about before, our initial goal was to do this for a year, and now I think we're coming up on a year and a half, or I guess 2 years here before too long. I think that's April would be 2 years. So yeah, we're definitely enjoying it, and thanks to all of you for also being a part of this journey. For updates, we have thankfully the weather seems to have chilled out. Things are going pretty well. We have fast tests going out every day. Sex determination should go out a couple times this week again. And then the panel tests, those will be getting run this week through the week. So I expect some results to start going out over the weekend, hopefully by Monday at the latest. And then a reminder, this will be the first time we're doing the 15 new tests. So these will be 45 morph panels instead of 30 morph panels we've always done in the past. So bear with us if we have some difficulty. There's going to be 15 more things that I have to make sure work properly on this one. Of course, tested it a little bit here and there, but this will be the first time we're doing it on a whole bunch of tests for people, or a whole bunch of samples for people. So bear with us a little bit if we need a little extra time to get all those 15 new ones out. But that definitely very thankful. It seems like you all are very excited for that, and we really appreciate all the samples coming in for that panel test. Let's see. I think that's all I've got for this. Kayla, how are you doing?
Hey, Ben, I'm doing great. And it's really, really cool that you're able to do like mass orders of 45 genetic tests all in one. And yes. I know you have to take like an individual sample for each one of those tests going into a panel test, right?
No, that's the cool thing. So the process that I do here in Virginia for the panel tests is very different from what they do. What you're thinking of is how they do that in Texas. So if they're doing tests, if we want to be able to get you results in 1 to 2 day turnaround, then yes, every single one has to be done individually. For me, the reason why we can't add the brand new tests to the panel right away is because I have to get them. So like in this case, I had 30 that were working well together. Now I've got to add 15 more and make all 45 work well. So sometimes the 30 are working great and I add one more and then 2 of them drop out and I have to, So it's kind of like, juggling 45 different things and having to make sure they all stay up. So
no pressure.
So yes, the very first time doing all 15 for many samples, we might say, hey, sorry, this test or that test didn't work at all. We're going to have to run those as fast tests. I don't know for sure what will happen, or maybe this time, since we've done it as long as we have, maybe I'll nail it the first try, but just give us a little space if we don't.
Yeah. Fair enough. Well, I'm excited for this episode because we're into talking about colubrid stuff, which makes me happy. And of course, all of our colubrid audience who have been waiting for years following our work with ball pythons. And we're still doing ball pythons, of course, and continuing to work on those. But also, it's cool to add more species into the mix. So let's knock out some business stuff, then we'll get to it. So firstly, we have a blank page, and then next is sheds that we want for ball pythons. They are the same as last week's. We have been getting a lot of shed submissions from people who are helping with our research, so y'all are awesome. These are definitely ones that we are aiming for right now, so if you have them or friends who have them, that would be great. in order to send sheds our way, just make sure that you're sending a little patch. You don't have to send a whole shed. And the species, animal ID, known morphs, possible morphs, lineage, and who produced the animal. All of that helps us tease out sometimes the little mysteries that pop up in these while we're figuring it out. Sometimes it's short and simple and very easy, but other times we end up with things like I don't know, monsoon. Honestly, just become a meme at this point. Not because he's the one doing the work, but for me, who just like that. Anyway, yes. Where was I going with that? Anyway, so we do have a spider complex panel. If you haven't heard, so that one is. So is that going to be going out at the same time as the newest panels?
Yes. Yep. They'll be run each time. When we do the 45 morph panel, we'll also be doing the spider complex panel. So,
and of course, as we mentioned, the 45 morph panel test, we added 15 more tests to this massive panel and only increased it by just $9. So it is a really good deal. It's like $3 and I think 8 cents per test, which is amazing. So if you have, you want to know what's hiding in those hets, this is the most bang for your buck to figure out what your ball python might be hiding. You also have morph testing bundles. If you don't want to do a full panel, you can do select a la carte, hey, I want this, this, and this, and get a discount for adding, for bundling those together on both of our vendors, Morph Market and Clutch. So they're all awesome. And we, the more tests we're able to do all at once, it's actually more efficient for us both and more cost effective. And if it's more cost effective for us, we like to pass that on to you guys. So,
and that's why we were able to drop the price down to $40 for that base price for a single morph test. That's because of you also, your support. We thank you once again.
Definitely. All right. And another thing we wanted to mention was the very first episode of the Herb Collectors Network. And as you can see, there's a whole bunch of people who have been on this podcast actually on that thumbnail. And all of these guys were talking about their very first episode was the acid ball python morph. Both the history, the weird genetics and defining traits of acids. So it's just really, really cool stuff.
Yeah. And they've got Josh on there, the person that worked with it off the bat, proved it out, and some of the story behind that. So that's pretty cool. So yeah, Morph Map, I think right now they're planning to do it about once a month. It's pretty cool, pretty good information. And Tommy, that's kind of running or heading up that specific Morph Map episodes. She also in her social media gave, for those of you that are looking at ball python morphs and maybe you're new and you kind of hear some of the terms like alien heads and floating this and that and flames. So what she's done is she's gone in and made some really nice, simple graphics along with a specific description. So Inky Clouds, if you go to her Instagram or Facebook, you're able to see these descriptions. She's done a really beautiful job of making it simple, something you can look at the picture and read the description. Really glad Tommy's doing that. I'm sure she's got more of that coming, but that kind of goes hand in that morph map episode they're going to be doing every month.
Definitely. And honestly, like I really liked reading into those myself because like that's honestly that has some applications like when you're talking about other species as well. Not necessarily the alien heads, but like blushing versus what are some other terms she used? Like blushing, flaming, banding, etc. Like some of it's fairly self-explanatory, but if you're fairly new to it, it is like drinking information through a water hose if you're trying to learn what mutations these reptile nerds have been talking about for decades. So it's good stuff. Definitely worth checking out. But yeah, let's talk about corn snakes. Here, I will go ahead and bring on our guest, actually. This is the wonderful Sarah Mohr of Sarah's Snake Shop. Hi, Sarah.
Hi. Hello, how are you today?
Good.
Glad to have you on.
Awesome.
Yeah.
Glad to be here.
Yeah, like your little friend that you have with you.
Thank you. This is Nectarine. He is a Halo Snow corn snake. I hatched him I want to say in 2017. I can't. No, his dad hatched in 2017. He's a 2020.
Okay. Oh,
Yeah,
he's
he's a
very pretty.
2020. Thank you.
Yeah.
He's very mobile today. But I don't know if you can see some of his colors kind of up close.
Yeah.
Maybe not. He's got some yellows coming in there.
Yeah, I see the
yellow checkers on his belly.
pretty.
And,
I love bright yellow belly checkers.
Thanks, I do too. He's he was the meanest snake in the bunch, and that's why he didn't sell. And so now he lives with me.
I love it. Honestly, the
But
spicy
now
ones.
he's just chill. Yeah, now he's just chill, and he
Oh.
has sired many clutches for me.
What a man!
What a man!
That's awesome. So just to preface this, so I mean, of course, ball pythons have kind of been our thing from like for the last few years. It's more what we're known for. But now that we are branching into our first colubrid tests with terrazzo, scaleless, it says albino, but the corn snake world calls it amel for this specific flavor of albinism, and lavender. And we've also had colubrid sex determination, but as far as morphs go, we're glad to be able to do that. And we figured of all the people we could bring on to talk about it, Sarah is definitely one of the people that we really want to talk to about this. We did mention a little bit of our plans for doing this, like all the way back in, I guess it would be 2022 now, when Ben went on to Snake Discovery to talk about colubrid sex determination testing and his goals for doing morphs eventually as well. And so here we are 3 years later and we're doing it. So it's really nice. Ben, did you want to say anything for that or?
No, that's great. What I'm looking forward to hearing is a little bit about Sarah. Like, how did you end up wanting to be able to touch and keep reptiles, and then why specifically corn snakes, and then why specifically the morphs that you've been working with?
Yeah. So my love of reptiles is literally from the crib. I have a photo of me with a like 5-foot red-tailed boa in the crib as a baby.
Gosh.
Obviously supervised. However, it has been in my blood forever. My grandma always she had snakes. My dad had snakes. And so I grew up with catching garter snakes in the backyard. And that actually was my first like real interaction with snake reproduction. My dad had caught this big old garter snake, and we just put her in a 10-gallon tank. It was just going to be overnight. You know, I was 3 years old. He wanted me to watch the snake eat, just experience it as a child. Snake didn't want to eat, but the next morning we woke up to about 12 snakes instead of having just one.
Awesome.
And so I was able to sort of, I was introduced to snake reproduction at a very, very young age.
Yeah.
A garter snake having babies in my bedroom at 3 years old.
I'm jealous.
I mean, who would have thought? Like, never in a million years. But thinking about that, like, we would have had a ton of baby garter snakes in our backyard the next day, either way, which would have been crazy. Regardless, so I've grown up with snakes, and corn snakes specifically, that started when I turned 18, and I got a job at a pet store that was local. And I saw my first snow corn snake, which was a pink and white snake. And, I wasn't a super girly girl, but the idea of having a pink reptile was just like insane to me. And so technically my very first corn snake was a snow corn snake. It did escape because I was young and dumb and thought that corn snakes could be kept like garter snakes, and they cannot. Corn snakes are much more Houdinis than garter snakes are. Garter snakes are just like, whatever, I exist here now. But corn snakes want to get out. And so I lost the first one, but then I got what I consider to be my first corn snake, which was just a normal, and his name was Flair. And I still have his grandchildren and great-grandchildren and great-great-grandchildren. He actually passed away a couple years ago. He was really, really old. I mean, he was old. And so he lived a long, happy life. He passed on much genetics. And so anything from my Flair line is from him. And then, but he was not the first one that I bred. When I worked at said pet store, I was usually a cashier, but I often worked in the reptile area. And someone came in one day and just was like, I have a snake. Do you want it? And I was like, well, what are you going to do with it otherwise? And they're like, we're just going to let it go outside. And
No.
I was like, please don't do that. And so that day I acquired a gigantic amelanistic male corn snake. And his name was Big Bo, and he was the biggest sweetie ever. He was morbidly obese. He was in a 10-gallon enclosure, even though he was like 5 feet long. I don't know how he was still alive.
Poor baby.
But he also went on to sire many, many snakes, including, I think he's his great-grandfather.
Oh my gosh.
Yeah, so I still have Bo's genetics as well. And I ended up breeding Bo to 2 females that I was told were males. And at the time, it was a drop off. I was the drop off place for corn snakes at this point. And so people were just like, here, I have 2 male corn snakes. I've always housed them together. And I only had 1 big tank. And I was like, I can house them together for like a couple of days. And then to get like everything set up, right? Well, 2 months later, I had 2 females laying clutches of eggs. And that was like April of 2011. And that July, I believe, is when I was hatching my first corn snake clutches, which included his great-grandfather hatched in that first clutch. So it was Bo was his great-great-grandpa. And so
my goodness.
I still have that line, and that's where Halo came from. So the original Halo came out of for me at least, and this is his dad. This photo is his dad.
Okay.
So he is actually more impressive of a halo to me. It's the yellow around the borders. But he can't sit still for photos. So his dad is the photo that you have. That's Frangipani. That's Frangipani, and his dad was Prince, and his dad was Bo. And Prince was the first halo corn snake to come out of my collection. And I knew he was different because I hatched 2 snows. Prince and Popper is what I named them. And Popper was the mean one. And he's the one that I sold off. And a year later, the people came back to buy rodents from me, and they brought him along just to be like, oh, hey, here's the snake we bought from you, just to show you what he looks like now. And he looked like a normal, regular snow. But my boy had developed these bright yellow borders that just were like neon yellow. So, and I think at some point, I believe it was around 2012. It must have been 2012. I posted photos, comparison photos online, and Don Soderbergh saw them and was like, hey, you should test that yellow to see if it's a mutation. I don't know if anybody, probably people who know me know of Don Soderbergh. He's a pretty big name in the reptile community. And so I was like, well, if Don says that this is something that could be something, then I'm going to test it. And so I spent a decade after that testing it to wild caught, like breeding a halo snow to a wild caught female, breeding the babies back together, do the whole shebang of testing to eventually come to the conclusion that it is incomplete dominant. And it's also, this is the heterozygous version of what we call green blotched snow, which I did not send you a photo of. But if you look at this halo and you see the pink areas, on a green blotched, those are just solid yellow. Instead of it ending at that border, it goes all the way through the whole saddle shape.
That's cool.
So, and green blotched had been around for a long time, but nobody really messed with testing it, I guess. I don't really know what was going on because green blotched was around way before 2012, way before I started testing it. So, but either way, I think it was actually 2022 is when I kind of put my mark on the world as the one who proved halo and green blotch to be kind of an incomplete dominant mutation.
Yeah, that's cool.
Kind of my thing now.
Well, that's amazing.
Thanks.
Yeah,
I worked hard on it. And then after that, I think it was a few years later. It was like 2016. Joe Pierce, who was working on the Golden Project, which is still one of the most elusive projects to this day. It is actually 25 years to the year that the very first golden corn snake was caught in the wild. So I believe that that's correct. Or is it 20 years? It's either 20 or 25 years. I looked at it the other day. I couldn't believe it's been that long. But Joe Pierce had produced some. He was testing it with caramel because there's another mutation that looks very similar. Proved it to not be compatible with caramel. So we know that it's its own separate mutation. And of course, you know, when you have two mutations like that look so similar and you breed them together, you have a bunch of normals because corn snakes, almost everything is recessive. So you end up with a bunch of normals that carry two mutations that look almost identical. And Joe Pierce just didn't want anything to do with that. And so he sent me all of those babies. And so everything, for the most part, that I have as far as trying to get golden going is like from the golden caramel test. So anything that I hatch that looks like that could be one or the other. And that's why I personally need you guys to come up with a caramel test. Because I want to be able to send you some sheds and be like, okay, is this snake a caramel or not? Because if it's not a caramel, then I know it's a golden, you know? And there's only, as far as I know, so Don Soderberg and Travis Whistler both have a chunk of this project because I ended up sending a chunk to them. Because I also ended up with a ton of normals. Like, what do you do with 1 million normals, you know?
Right, yeah.
So I sent a bunch to them. And Joe Pierce, I believe, is still working on it. Last I heard, he had hatched quite a few, like 2 or 3 years ago. But he tends to drop off the map and nobody hears from him for years. And that's where we're at right now. Nobody knows what he's doing. And then there's me and then like one other person who are working with it. And between all of us, maybe one day we will actually get this mutation off the ground after 20 something years. So even though it's one of the longest like standing mutations, it is impossible to like actually get going. For some reason, it has taken over 2 decades to get this mutation off the ground. But this is, I believe, like an F3, the photo that we have here. So, I mean, this has been from the wild caught. And so, I mean, obviously some breedings have been successful. This is Joe Pierce's photo, but it has just taken forever. The babies don't tend to thrive very well. The females tend to have fertility problems. Like there's a lot of, and there's also an unfortunately high number of dead and egg babies from this project. So we're hoping that with some outcrossing, some of those issues will go away. But first we have to get visuals that will breed. So
makes sense.
That's where we're at with golden right now.
Man.
And so the most recent one to come into my projects is Weirdo. This was found it was found out of two what we call tessera siblings, which are like tessera offspring that are not visual tessera because tessera is dominant. But not non-visual tesseras that they just look really weird. So non-visual tesseras that were coming out of the original tessera breedings just had this very strange look to them. And a couple of those were eventually bred together by Catherine Turley, and this weirdo looking probably mutation came out of it. And there's not too many people working with it, but I know Ryan Farrell of Farrell Captives is one of those people. And the snakes that he produces, I'm pretty sure it's either dominant or incomplete dominant, because he bred, actually the male on the bottom of this particular photo I purchased from him, and he bred that male to a couple of like non-related females. And the babies look like this now. Like they're a year old now and they look just like this. So either it's dominant or maybe it's incomplete dominant. Don't know. But that's kind of one of the newer things that we're kind of working on. I say I'm working on, plus a few other people. Not too many people have been interested in it enough to really work with it. So I'm glad that both me and Ryan have enough interest to like be at least trying to figure it out. And I really... Unfortunately, there was another line called Weirdo before this Weirdo came into existence. And so we're having a bit of naming issue because now we have two different things that are named the same thing, which has happened before. We had strawberry. Strawberry was a big issue we had before because people were using it for both the strawberry mutation and the red factor mutation.
Oh no,
and it took a long time for people to separate those two. And so now we have a weirdo line, which is a very special Miami line. I believe from Midnight Exotics. Maybe it's the same person who has the Tessera photo that I sent you guys. And their name's on there. But they have a weirdo line that predates this weirdo line. So I don't know what we're supposed to actually be calling anything at this point.
Weirdo and weirder.
Weirdo and weirder. Yeah.
For real. Yeah. I've just been going by Turley's Weirdo. I've been calling it Turley's Weirdo because Turley is the one who hatched the first ones. So that's what we're calling it.
Yeah. And so, I mean, speaking of like naming and figuring out like where these come from, I guess that might have been part of what inspired you to write your books, right?
Yes. Yeah, absolutely. So I have forever been obsessed with the histories of things. So I love like etymologies, like of words.
Yes.
And it's been that way about everything. I just love knowing the history of things, just literally anything. And I started typing up for myself all of the histories of the corn snake mutations. And I had a protege who would come over and help me clean snake poop, frankly. But he was learning a lot about snakes, and he's actually a marine biologist now. I'm so proud of him. Yeah, he started with cleaning snake poop in my basement, and now he tracks sea turtles that are endangered. And shout out to Trevor Proctor. He's one of my favorite people. He'll probably eventually see this.
Trevor.
What part of the ocean is he tracking them? East Coast?
I'm sorry, what?
What part of the ocean?
He is, I want to say it was, I don't know, it was some island over by Africa, I think. I'm not 100% sure. I'm so sorry that I don't know the answer to that.
That's okay.
But it's somewhere far away, somewhere in the like literal jungle.
That's awesome.
Yeah. So I had printed off a copy of my typed up corn snake thing for myself, just so he could learn about corn snake morphs. And he's like, you should make this into an actual book. Yeah. And so that's when that kind of started. I did not know how to write back then. And so the first volume, you can see, or edition, you can see the volume 1 has an edition 1 as well. Edition 1 is relatively poorly written in comparison to edition 2. So you don't really want edition 1, but I just did it. And I did it self-published, and I printed them all myself for the first, I think I printed 100. And it's about all the histories of all the corn snake morphs, where they came from, what the actual mutation like does to the animal. Like, we obviously have things like amelanistic and anerythristic, which take away certain pigments. But then we also have things like red factor, which enhance pigments. And then we have like stripe and motley, which is, pattern mutations. And we also have scaleless and microscale, which are scale mutations. And we also have lavender, which is kind of an albino type, but it's also kind of a scale mutation as well. We have a mutation like that in humans also. I probably should have sent you that scientific paper, but I don't know where it is. I think I have it in my lavender deep dive video. So there you go. There's my YouTube channel, youtube.com/Sarah Snake Shop. See, I did the same thing that you did earlier.
There you go. Oh, you're fine.
And I have morph deep dives on almost every single corn snake mutation.
Yeah.
For like individual things, what they do. I also have deep dives on what each mutation looks like when it's mixed with other mutations. I'm still working on that whole process, but I have a few videos up in that like series. Yeah. So lavender, the mutation affects, I believe, the way that pigment goes, like it affects the keratin. Hold on. I know that I have it. It's in my lavender video in the description. So that's one that if you would like to specifically go to my channel for any one video, that lavender video has the information on that mutation in humans and how it is actually technically a form of albinism, even though you can tell it does have some pigment. Now, the reason we know that it's The reason we knew it was a scale mutation before we knew
Mhm.
about a similar mutation coming up in humans, like we knew there was one, but we didn't know exactly how it worked, and we didn't know if it was the same thing. Now we do, kind of. You know, obviously snakes and humans are different, but it's a similar mutation. We had scaleless lavenders. That's how we knew. So we were breeding lavender into scaleless, expecting to get a bright purple scaleless baby. But instead we just got a normal looking baby.
Hmm.
And
Interesting.
so scaleless lavenders kind of just look like normals. They kind of just look like normal scaleless. And so that's how it was kind of tipped off that like this mutation is not happening in the pigments of the skin. This mutation is happening somehow with the scales. And then come to find out it's a mutation. Humans that have this mutation have like silver hair, like gray silver hair from the time they're born.
Ooh.
Yeah, so I can't remember what it's called, but it's out there. It's a form of albinism.
Dude. Okay, yeah, no, that's gonna be a fun rabbit hole to go down. That's cool.
It was so fun to research.
Yeah. Oh my gosh.
There's definitely specific genes when they're mutated in a mouse or a rat or an axolotl or zebrafish or human. You know, a lot of the albinos really come down to like 4 or 5 different genes where there's a mutation.
Yeah.
So like the one in corn snakes is also the same as one of the albinos in ball pythons, and the same as albino in humans and in, you know, mice, all that kind of stuff. So yeah, there's definitely some similarities, and it's very interesting. Obviously, a sign of, you know, the evolutionary process where something as different as a human and a snake, you put a mutation in that gene and it will give you a similar look. You know, that's pretty.
Yeah.
Too cool. And let's see. So these are just like, so as far as scaleless, I put these up since we have a scaleless test too. When you mentioned lavender scaleless, I was like, oh, I wonder if, you know, this little guy right here is like a lavender scaleless, but apparently not.
Nope. The one on the bottom right is more what a lavender scaleless looks like.
No way.
It's not exactly that. It's not exactly that, because that's an amelanistic scaleless, so that has no melanin.
Right.
But they are a little bit lighter than a normal scaleless. They do kind of have a slight like hypomelanistic effect. It's not like normal scaleless. So like technically we could say that lavender is a hypotype, but it's also an albino type. So all terminology. It's all terminology.
Oh yeah.
Technically,
nature doesn't like to give it to you straight.
Right. Oh yeah. Well, yeah. Nature's not going to be straight. It's just going to be what it needed to be to survive.
Exactly. Or what survived the selection process that we did as breeders.
Yeah. Exactly.
Let's see. So is there like, since you wrote the book on corn morphs, or one of the books on corn morphs, are there?
One of them.
Yes. Because I know Kathy and Bill Love did a lot of really cool stuff that way too.
And Charles Pritzel had a whole series, which is essentially kind of what I wanted my books to end up being, like kind of replacing a little bit. He had a whole series up to 2012 of all the updated corn snake morph information. So, and of course, 2012 is like right after I started, and then just like nothing new came out, even though we knew new mutations were coming out. Nobody was writing about them or where they came from or anything. And so not only was I attempting to like learn myself about the new stuff, I also wanted to help other people learn about the new stuff. And these that you're showing are the ones that you guys currently have tests for.
Yeah. Ben, did you, like, while you were doing this, like, was there anything particularly interesting? Like, were there any, like, you said that I think the AML was the same gene as one of the albinos or same mutation site as I think you said the lavender albino in ball pythons.
I believe that's correct. I don't remember off the top of my head, but it's definitely one of the albinos in ball pythons.
Yeah, that's cool. Genetic mysteries are you interested in, like, going forward from here? Like, what sort of things are you hoping that genetic testing can help solve in the world of corn snakes?
Yeah, so there's a few different things. There are a few mutations that are suspected to be hybrids.
Mhm.
And I know that hybridizing is really weird, and especially when you're crossing it back for a few generations, it's really hard to detect any of that. But there is a suspicion that Tessera might be a hybrid with Caliking, because Caliking has the Newport morph. So this is a Tessera. Mystic Exotics. Mystic Exotics is the one, not Midnight Exotics. Mystic Exotics.
Gotcha. Yeah.
Now, I in no way am implying that Mystic Exotics is working with hybrids. I just want to say that right now. First Tessera photo that I had on my computer. So this is what a Tessera corn snake looks like. This one specifically is a very nice Miami Tessera with that silver background color.
Oh, yeah.
But it's suspected that Tessera may have originated with the Newport Caliking morph, which, as you can see, looks somewhat similar.
Yeah.
Now, that's again, it's one of those genetic mysteries we just don't know. There's also suspicion that the Ultra mutation was originally from black rat hybrid. Now, I think that's been debunked. I heard an interview with someone relatively recently. I can't remember who it was or where it was. I think that he did debunk that this is not a hybrid mutation. But since the suspicion has been out there since, this mutation came into being, something like that would be nice as far as genetic testing goes.
Yeah.
But something that's
Morph Market, I think it's still like in their Morphpedia, I think it still says the Ultra from across.
Yeah, and we still are not 100% sure that that's a fact, because the people who produced it originally, were just like, yeah, we don't know. It might have come out of our pure corn snakes. It might not have. We don't know.
Oh, no.
That'll be good.
Yeah, it was kind of unknown.
Interesting.
But so again, just having something that could test for any type of hybridization. Of course, scaleless is assumed to be a hybrid as well, any scaleless. So that kind of stuff. But I think that the longest standing genetic mystery in corn snake that we have had is our Toffee and Buff, the same mutation. So this is Toffee. Toffee was actually the second of the two mutations to be found. And then Buff looks very similar. You can see this is a little bit darker, but so the Toffee came out of the same group of snakes that Buff came out of. And those group of snakes all originated from Rich Zukowski's collection, because he was the first one to produce the caramel mutation. And these other excess yellow dominant mutations, both, assuming they're different, both came out of the same like caramel mutation. Or like that same line. Now, the yellow jacket mutation, which is a different one, is also believed to have come out of that line. I didn't send you a picture of one of those because that's it's kind of a little more isolated. And it's a little bit more, in my opinion, a little bit more. I think there's even more proof the yellow jacket is its own separate thing than Buff and Toffee being their own separate things. But they all did originate, as far as we can tell, in the original caramel lines.
Interesting.
And so I think that it would be really neat to know, like, to have a genetic test for one or the other or both, because they're both dominant mutations. They both kind of give this yellow wash over the snake, and they kind of reduce those reds. And when you look at them side by side, people who work with them do believe that they're different. And so I'm inclined to believe that they are different, because a lot of times when you see things, face to face and you're hands on with it, you can see those little differences. And, that's how people could tell the difference between like a regular hypomelanistic and a lava. Like, they're both technically hypomelanistic types, but they're just different enough for people to go, we're going to test that and see if it's different. But it's so difficult to test these two because they look almost identical and they're dominant. And it's almost impossible to identify them versus normals as hatchlings. And so people just don't want to mess with it. They're just, they don't want to mess with it. So I think that is like the most, I don't want to say it's the most needed, but it has definitely been needed the longest genetic test. I think that's just the longest standing needed genetic test. Is it the most needed now? I would say not necessarily. I think that finding out if tesseras, if people could find out if their tesseras were homozygous or not, I think that would be really valuable to a lot of people. So this is my subadult female palmetto. Her
Pretty
name is
girl.
Miranda. I don't know if you can see her little mask, her little bandit mask on her face.
Oh.
She came from one of the like original pairings. She's moving. Oh my goodness.
Yes.
So she's from Travis Whistler Reptiles, who did a lot of the work on sort of making sure that this morph continued, because Don Soderbergh is kind of slowed down, so most of his collection went to Travis. And this girl was one of the first that Travis produced, like in the first few years of those palmettos. So the reason I wanted to talk about palmetto is just because it's like one of the few like kind of incomplete dominant mutations we have. Like we have tessera, which is dominant, and this is a kind of a weird one for me. So obviously this girl doesn't have any bug eyes, which is good. You don't want those. But these guys do tend to get bug eyes, and it's because the mutation that causes palmetto also, like the what's the word that I'm looking for, the gene that gets mutated also controls how many cells go into the eyes. And so when you mutate a gene and it just mutates just a little wrong, it will push extra cells into the eyes and make the eyes bigger. So that's why bug-eyed happens. There is some evidence that, especially if you breed a male with bug eyes, that it will pass on more likely. But that's just anecdotal from breeder. That's like, that hasn't been like tested, but that's just from what I have heard from other people who have decided to breed their bug-eyed snakes, that it is more likely to happen in males, and males are also more likely to pass it on. So
interesting.
Take that for whatever you will. Most people just choose to not breed their bug-eyed snakes, and that's what I would generally recommend. I'm not really sure how much it helps, because it kind of seems like since you are mutating that gene, regardless, there's just a chance it's going to happen. There's just a chance that the bug eyes are going to happen. But another interesting thing with them is it was originally thought to be recessive, but it was found to be incomplete dominant because all of the het forms looked like hypos. And so I'm going to show you an example. I have two other females here of an anerythristic and an anerythristic het palmetto that are about the same age as that same girl. And I'll get them out at the same time.
Yeah, because I've read that it is incomplete dominant, but I've also had no idea like what's. Oh, I see.
You can see the lighter one here. This girl right here. She's het palmetto. You can see she's got a lot of light colors. She actually does have a lot of weird like peaches and yellows. And then this girl is just a typical regular anery. She's just kind of that black brown with a little bit of yellow, but not near as much, kind of up in that area there. And these girls were hatched the same year, so you can kind of see just the general difference of color and tone. A het palmetto is kind of in itself considered a hypomelanistic type.
Interesting.
Which I didn't believe at first. I was just a general non-believer. I was like, in my mind, I thought it was just probably because of the locality, because they just continued to breed, like an interbreed. And I was like, it could just be locality specific stuff that you're seeing here. But turns out, even after a lot of outcrossing, that's not the case. Turns out it is actually incomplete dominant. So there's kind of a good example of what that het does. It's more difficult to see in a normal, especially with my camera, but I thought in an anery it would be a lot easier to see. So there you go.
Absolutely.
Very cool.
That reminds me a little bit of like the like super rusty in black rat snakes, where it's more of an incomplete dominant, and then it ends up with this leucistic type of animal. And they're well, and a lot of people did think, kind of for that reason, that it might be a hybrid mutation. However, the official story is that it was wild caught in some lady's backyard, and that there were multiple of them. Oh,
wild caught in a
really?
lady's backyard. Yeah.
Wow.
So that's the story, and we're sticking to it.
Fair enough. Fair enough.
That's how it goes. But I can tell you all the things. I can tell you all the things that people have thought over the years in this hobby.
Oh, yeah.
There are things that are unprovable for the most part, unless genetics testing gets like super, super good for that kind of stuff.
Mhm.
And not only that, but you might need sheds from some of like the original snakes. So it's like we might need a shed from the first or second or third palmetto in order to find that out. We might need sheds from, when it comes to ultra, like we might need sheds from like the first or second ultras to figure that out. And so, it just is what it is now. The stories are what they are. So
yeah. Very cool.
Yeah.
And it's my job to tell the stories. Please go buy my books. They're available on lulu.com. I am the only one writing corn snake books on lulu.com right now, as far as I know. But I also have a 2025 corn snake calendar on there, which I personally use because the place that I normally I usually get a very specific I usually get the Savannah Cat rescue calendar because they do it every year, and since I own a Savannah Cat, I like to support that. But they stopped making their calendars, so I was like, well, I guess I'm making my own calendar, and I guess everybody else can buy it too. That's
there you go.
That way.
And you'll be at Colubrid Fest again. That's where I got your book.
Absolutely.
I got it directly from you, and I got it signed. I got a picture. So,
heck yeah.
So you'll be
I'll be there again with more books.
Yay. Sounds good.
I'm going to bring copies of the second book this time.
Yeah.
Yeah, I think I got your volume 2, version 1, volume 2.
Probably because I had the most of those. Yeah. I haven't actually done any printing of the second one, except for like a few that I just kept for me and like gave away to a couple people. But I will have the second book and the first book on the table at Colubrid Fest. Great. And Colubrid Fest is in my hometown. So if anybody wants any recommendations on restaurants, I know where to go.
Noted.
Excellent.
Yeah, no. Well, awesome. Well, thank you so much for joining us today.
Thank you for having me.
Yeah. I know this is going to be a huge treat for our corn snake people who have been following us, and we are just so glad to have had you on.
Thanks. Me too.
Yeah.
There's Bandit. That's the one that I just showed a minute ago. That's Bandit with her little face mask.
I love her little mask. So cute. But awesome. We definitely covered a lot of ground, and hopefully these will be something that we can, like, some of these mysteries are things that we can help tease out over time with genetic testing. I know that with there have been several things in the ball python community that we've proven are different, and a bunch of things that we've proven are similar, like Hurricane, Trick, and Blitz. They're actually the same, but also the Axanthics. There are some that have proven out, and others that we've done both actually for the Axanthics. So, so that'll be really exciting. And again, if you want to help submit sheds, either for these projects or just a project you want to see, again, you can go to raregeneticsinc.com/help, and that way you can send in your sheds. There we go. So thank you guys. We really hope you enjoyed this week. And be sure to look in the description. I'm going to have links to Sarah's social media, to her books, and all the above. So make sure you like, comment, and subscribe. We really hope to see you next week.
Thanks for watching.
Yeah. And outro in 3, 2, 1.